Thank you everyone for joining. Welcome to CWA's June edition of the Innovator Spotlight. Today's topic is rapid detection, and we'll be talking to two companies, Diamidex and Universal Biosensors, who are currently innovating within this space. We do have polling that will be launched during this webinar, so please participate. And we also have a Q&A function at the bottom of your Zoom. So feel free to send us questions throughout the presentation and we can get to them at the end. I would like to say a brief thank you to our sponsors of this webinar series, Badger Meter. They have a family of technologies enabling faster insights in the industry. Badger Meter is an industry leader, including their innovative Blue Edge solution for real-time monitoring and system optimization. So thank you, Badger Meter, for being a CWA supporter. And thank you everyone for joining again. My name is Natalie Van Scyoc, for those of you that don't know me. I'm the Innovation Associate with the Cleveland Water Alliance, and I talk to companies all over the world every day with our innovation team, assessing their viability for potential piloting opportunities, and CWA manages these collaborations in the region and beyond. One of the ways we're able to do this is through our Smart Lake Erie watershed and testbed. So with our testbed, we're able to have faster insights in what's going on in the water. And CWA has deployed over 7,700 square miles of telecommunications coverage across the Lake Erie watershed and over portions of the Ohio River. Enabling the deployment of over 200 water quality sensors capturing over 1 million data points annually. This equips CWA to assist with the ability to trial and demonstrate the various technologies across the IoT lifecycle we see from around the globe and in our world-class testbed. As part of our work in IoT sensing and monitoring, which facilitates faster insights, there has been increased interest and opportunity in innovative technologies that enable rapid detection. This family of technologies includes portable systems for in-field sampling and analysis, as well as benchtop devices for faster insights in a lab setting. And with that, allow me to introduce you to today's innovators. We have Sam Dukan, who is the CEO and founder of Diamidex. He's a research director at CNRS, joined ENS ULM in 1991, and he has a degree in hydrobiology and a master's in chemistry, as well as a PhD in microbiology. He's collaborated with companies like Suez Environment and BASF, published over 44 papers, and holds 14 patents. He also served on committees for ANESES and CNRS. And we also have Alex Certoma joining us from Universal Biosensors. Through his role as Associated Director, he supports the R&D team in bringing new innovative products to market through market identification and initial partnering and distribution agreements. Since 2020, Alex has been involved in 9 product launches across the human health, wine, and veterinary industries. Prior to joining Universal Biosensors, Alex worked in financial services. Alex has his master's in applied finance and a bachelor's of finance and materials engineering. And with that, I'd like to pass it off to Sam to tell us a little bit more about Diamidex.
Okay, thank you, Natalie. So it's a pleasure for me to be here today. So I will make it as short as possible, and please feel free at the end of the meeting, to ask all your questions. So at Diamidex, what we are doing is to make the invisible visible faster, to make the world safer. And the idea for us is to be able to detect earlier microbes, and please change the slide. And the idea is to make the possibility for everybody to perform microbiological analysis and to be able to deliver the same type of result with the standard method. So it's why we are culture-based method, and we reduce the number of steps for each of these methods, and we make an automatic enumeration of the microbe of interest based on a technology that I will explain later. The idea at the end of the day is to be able to install the solution in the field or in a laboratory to deliver equivalent result of the gold standard at the end. So please change the slide. So today, if we check on the way to enumerate microbes, we have more than 75% of the microbiological tests that are performed using culture-based methods, which are very slow and give a very accurate result. So the issue is when you want to make a better quality control risk evaluation, and if you need to wait several weeks to obtain results, it's not possible, to control correctly the sanitary risk. So by consequence, you have a lot of alternative methods that have been developed since more than 30 years. But even if these methods are very rapid, they are not usually reliable to the gold standard, to the Petri dish, and to the Pasteur, method since the 19th century. So there are still a need to identify a solution that will be able to deliver accurate result, but earlier, in order to save money for the company and save life. Please change the slide. So to make it very short, to understand the issue, for microbiological in water, enumeration, you start with your sample and you need to filter your sample on a membrane. You put your membrane on a plate and you have at the beginning microbes that are not visible by eye. You wait that this microbe will divide, which will take a lot of time, from 1 day for the faster and several weeks for the latest one, in order to form colony. When you have this colony, it's just the beginning of the story, because after you need to identify the colony of interest, which need skilled people and more, technical steps. And this is what is done since the 19th century. At Diamidex, what we are doing is we detect, identify, and enumerate the microbe of interest at a stage of microcolony, and we do it automatically. So which means that we reduce the time to obtain the result, and we reduce the labor time associated. Please change the slide. So what does it mean? It means that we are manipulating deep science, but at the end, what we want is to make text that will be simple for everybody and at an affordable price. So we are located in France, and we are a team of 34 people that work on this topic since 2014. Please change the slide. So what we have designed is a MICA, which is a microcolony automatic counter. So it combines an equipment that is based on optics and mechatronics, some specific probe that allow to label specifically the microbe of interest, and a machine learning system that allow to make the difference between the right microorganism and the wrong one, the one that you are not interested to detect. And this technology is protected by 6 family patents, and it makes our technology the most powerful solution to enumerate microbes in a colony forming unique level and earlier. Please change the slide. So today we have 2 equipment, one that is based on fluorescence, and we are able to detect and enumerate Legionella pneumophila in 2 days instead of 10 days. So we divide by 5 the time. For water, we are able to detect and enumerate Pseudomonas aeruginosa in 1 day instead of 2 to 7 days. And we just released a new solution for Escherichia coli, where we enumerate E. coli in 6 hours instead of 1 day. One equipment can perform until 500 tests per day, which is a lot. And usually we reduce, like I say, 4 times the labor time. We have another equipment called MICA Highlight that is based on light. And this solution allows to enumerate the total viable count and divide by 2 the time to result. Next slide. So just to make it very simple, for Legionella pneumophila, which is our best seller. It was our first product, but has been released in 2024. So we use some specific probe that label specifically Legionella pneumophila. We take more than 100 images of the membrane after 2 days of incubation. And based on machine learning, we are able to make the difference between Legionella pneumophila microcolony and all the other microbes. Please change the slide. So just to give you an idea here, on the left, you have a membrane after 2 days of incubation. You see almost nothing except one big colony. And based on our solution, we are able to see what is detected by mica on the image in the center. You see a lot of spots, that are fluorescent. And with the machine learning, we detect the microcolony of Legionella pneumophila in the right, and we are able to count 166 CFU per liter of Legionella pneumophila. Next slide, please. So today we have a very we have accelerated our sales. So we have today 14 customers. We have 25 trial phase. And since last week, where I sent the document, we just sold one new solution in Switzerland, and we are on the way to sell another solution in South of Africa. Our distributor network grew very fast. We got 8 distributors in 2024, and today we have 17 distributors that cover around 25 countries. Next slide. So we believe that we will be the future gold standard of microbiological testing, and with our solution, you are able to react earlier in case of microbiological contamination. Feel free to contact me if you need more information. Thank you.
Thank you so much, Sam. That was a wonderful overview of Diamidex and your mica solution. I want to remind everybody again that there is a Q&A function in Zoom, and please use it if you have any questions that we will get to towards the end. And with that, I will pass it over to Alex to talk more about universal biosensors.
Thank you very much, Natalie, and thank you everyone for joining. Really excited to present universal biosensors in our solution Aquascout today. So next slide, please. So Universal Biosensors are a company. We're based in Melbourne, Australia. We've been around for 25 years now. We're a global biosensor company who have a long history, 20 years plus, in manufacturing consumable electrochemical test strips for use across a variety of industries. We're a world leader in electrochemical cell technology, and this technology has been used to deliver more than 15 billion tests to patients and customers worldwide already. We have a suite of 11 portable electrochemical sensing products currently across the human health, wine, and veterinary industry. And we are in the process of developing and finally launching a portable heavy metal sensor called Aquascout. Next slide, please. So Aquascout is a handheld water testing product which enables you to detect and monitor heavy metals in water samples in the field. We're specifically starting with lead and copper, and then hoping to extend the platform into a number of other heavy metals into the future. Like most of our tests, what Aquascout addresses is some key issues that are associated with the current major method of testing, which is laboratory testing, and is challenging the ability to provide results in a more time-effective manner and also at a lower cost. So the Aquascout platform combines 3 parts. You have the handheld portable analyzer on the left, which allows you to take the measurements. You have a consumable electrochemical test strip in the middle, and then you have a again consumable, a lyophilized or dried down buffer, which you add the sample to, and then you insert the test strip and allows you to take a measurement. There's a number of potential market opportunities that we have with the Aquascout product, but our initial target market is specifically drinking water utilities and helping them in their lead pipe inventory and replacement. But other opportunities as we add additional metals to the platform can be in, mining, industrial waste, potentially water compliance as we move towards things like EPA approval, but then also the potential to allow consumers and other interested parties to test their water as well. Next slide, please. So in terms of the initial target market, as I said, what we're most interested in at the moment is helping utilities with their lead pipe inventory and replacement. So as I'm sure you're all aware, in the US, utilities have until 2033 to remove all lead service lines and galvanized pipes in their network. There's a variety of current techniques that are used, which include anything from visual inspection, full excavation, or water analysis using laboratory-based methods. All of these have their positives and drawbacks. A lot of them are very time-consuming, costly, or require trained professionals to perform them. This is a really massive undertaking, as is an estimated over 9.2 million lead service lines, which are still in the US, and they're quite heavily weighted towards the northeast, southeast, and midwest of the country. What's even more interesting is that on average, water utilities in the US have over 30% of their service line network, which is currently made of an unknown material. And there's no easy, cost-effective way to be able to identify a lot of these materials. And that's really what we're trying to help with. Scyoc, we hope, can offer a potential solution to this problem, but also have potential to be used in conjunction with some of the other methods as well to increase the opportunity and basically increase the chance that you make the right identification of the material. One of the other things which we've found out over the last couple of weeks, and a potential opportunity for using this by utilities, is in dealing with customer questions. So we talked with a utility a couple of weeks ago who, following publishing their inventory, had hundreds of customers calling them asking about their pipe, which was made of an unknown material. This caused them to have about 4 weeks backlog of lead testing in their lab. And we hope that using a product like Scyoc could help, easily service this issue and also provide a much better customer experience. Next slide, please. So how it works is Scyoc detects your metal of interest in water by using an electrochemical sensing technique called anodic stripping voltammetry. What we do is we basically apply a certain voltage, which coats the gold test strip with the metals. We then apply a reverse voltage, which strips that metal off. And then we measure this current difference, and then we can associate that with the concentration of the metal that we're trying to detect. What makes these tests specific for each metal is that each of them has a different buffer, which we can use to produce an electrochemical signal that's only gathered from that metal of interest. And the sample test workflow is shown below quickly. So basically, you can select which metal you're trying to detect. You can then put in a sample ID to show what you're testing, whether that's an address or a pipe number. You then select the buffer index that you're using. This just helps us identify which manufacturing lot of the test you're using, so we can make sure that you have the right calibration parameters. So no matter what you're testing with, you're going to get the same result. You then just need to add 6 ml of your sample to that buffer vial. The test strip then gets inserted into the handheld analyzer, and the test strip vial with the water in it then gets screwed on to that analyzer, and the test gets completed within 3 minutes for lead and 30 seconds for copper. So we can provide quantitative measures for both. With lead, we can sense between 5 parts per billion and 100 parts per billion, so sense quite low down. And with copper, we can sense between 50 parts per billion and 2,000 parts per billion. All those results are shown on the screen. They are also able to be sent wirelessly via Wi-Fi or a wide connection as well, to help utilities manage their data as they start to collect this and input it into other systems that they may have. Next slide. So some of the overall benefits that we have compared to traditional lab testing obviously include the near immediate results. You're talking about max 5 minutes compared to, days, sometimes weeks if you're sending a sample off-site. The ability to test in the field, particularly for something like customer complaints or customer questions, can be very helpful. A really easy to use workflow where anyone can do a test. You don't need to be trained professionally or as a chemist to do this. Your results can be transferred wirelessly, and obviously we can be much lower cost. So the handheld analyzer we're looking at selling retail price for around $2,000, and then the test per test, which is one test strip and one buffer vial, is going to be around $10. Another really important factor is giving utilities the opportunity to control their own testing if they don't have their own lab, which allows them to do more testing if they need it, or more targeted testing instead of sending it off-site. And then we're striving to be obviously comparable in accuracy to that lab method. So we've gone through some initial field trials where we show really good correlation to ICP-MS measurements, and we're just going through a few more in the field to really increase that performance and narrow down to be something of comparable to what's currently done. Next slide, please. And so where we currently are is all the base development of AquaScout has been complete. We're currently going through a number of sort of pilot trials in both the US and the UK to really understand the performance and usability of the product. We currently have 2 sites up and running with the hope that we can get another sort of 3 to 4, particularly across the US, involved in this. Following that, sort of in that September to November period, we'll be implementing any required changes that we identified through these studies, mostly of the testing algorithm and any software improvements. And then in the background, what we're doing is setting up a full sales and marketing structure to support the product launch, with the product initially targeted to launch December this year, January 2026. So really the 2 big things for us at the moment is trying to get as many utilities as we can using this over the next sort of 3 to 4 months to get as much feedback as we can before we finalize the development. Obviously, this can continue as we move forward through to commercialization, as we understand that piloting and trialing these solutions is very important in this area. And then we're also in the process of talking with potential distributors and commercial partners to provide an avenue for us to get this into the hands of utilities in the US going forward. Next slide, please. So yeah, in summary, we're coming to the very end of this development. We've been doing it for 18 months so far. We've got another 6 months to go. So really excited by the potential of this solution, and happy to answer any questions, as well as Sam, obviously, after in the next sort of 20 or so minutes. Thank you.
Thank you so much, Alex. That was wonderful. Yes, please use the Q&A function if you have any questions for either of these innovators, because we will be pivoting to the Q&A panel discussion now. So I'd like to stay on Alex for a second. How do you educate your buyers and end users about what becomes possible in their work as a result of using your technology?
Yeah, I think a lot of it is to do with the marketing efforts and outreach that we're starting now, trying to demonstrate, the costs and the impacts of how it's currently done with an external lab, and really demonstrate the benefits of being able to take this in field, being able to respond to questions or queries quicker, and really helping them understand, I guess the legislation, government push around the whole lead pipe removal as well, and what they're going to have to meet, because not everyone understands, I guess, the massive undertaking, or how massive the undertaking is going to be, and that there are solutions out there that are going to make it hopefully a little bit easier.
Right, definitely. And Sam, I'm gonna ask you the same question. How do you educate your buyers and end users about what becomes possible in their work as a result of using your MICA solution?
Wow, it's not a simple question, because we are focused on laboratory So we are not in relation with the end user, you know. So what I can say is that our buyers are interested by our solution because they increase their productivity, you know. They are able to perform much more analysis, you know, with the same period of time and with the same number of technicians and same number of equipment. And in the meantime, they are very happy because they can deliver premium result to their customer, you know. So this is something very important for them.
Yeah, totally. Thank you so much. And we had a question in the Q&A for you as well. Is the mica equipment primarily for use in the laboratory, or can it be adapted for continuous water monitoring?
No, it cannot be used for continuous monitoring, but it could be installed in the field. We have a customer that have cooling tower, you know, and that have installed in the facilities our solution. And in between all the regulatory analysis, they perform mica analysis for Legionella, especially, and they are able to adjust and adapt, you know, the control and the biocide and all the molecules that they add in the pipe, you know. So it's a way for them to adjust, you know, the treatment, which is very important.
Wonderful. Thank you so much. And that is
Thank you.
absolutely crucial. Alex, we have a question in the Q&A for you. What makes your approach superior to other wet chemical approaches for lead detection?
I think I guess it's a combination of the usability and also the accuracy compared to lab-based methods. So I think a lot of the other rapid methods might not necessarily get down to the limits of detection that we can in the field in a portable product, but also then closely align with what you get in the lab. So that's something that we've been really focusing on. And one of the other reasons why doing these initial studies are really important to us is to make sure that we are really, really accurate down in that 5 to 15 parts per billion range, because that's where most people are going to see their readings if they're not less than 5, which hopefully they are. And so I think that's sort of the main selling points for us is ease of use, but also being as accurate as what you can currently do in a lab.
Right. That's wonderful. Thanks for that insight. And I know earlier you touched on how the EPA guidance has been focusing on lead service lines and all of that. Could you speak to how UBI's technology applies to the efforts and how these regulations are shifting how you are marketing?
Yep, absolutely. Yeah. So I think there's sort of two main uses that we see for the product. There's one as a screening and complementary to what's currently done, which means that you're not providing an EPA approved result for something like compliance testing, but you're finding a much easier way of being able to identify where problem areas are, or if, again, customers are calling in for asking you to test, that you can go out quickly in the field with any operator and do a quick test. The other area that we're also investigating is there is an EPA-approved method which this product would lie under in a laboratory, so potentially also working with labs to provide them with a more cost-effective solution than running, large ICP-MS to provide a EPA-approved or compliant result as well.
Wonderful. Thank you so much. Shifting back to Sam, not everyone may be familiar with Legionella, so could you speak to the importance of receiving quicker results and how it impacts the community regarding the dangers of Legionella exposure?
Wow. Okay. So, I mean, indeed, maybe not everybody is familiar with Legionella. And what is important to have in mind is that you have probably in US 20,000 people that are infected every year, by Legionella. Legionella is growing on hot water, and when you generate droplet of water coming from cooling tower or just from your shower, you breathe this droplet and it goes into your lungs and you become sick, and around 10% of the people that are infected, die due to Legionella disease, which is Legionnaire disease because it came from Philadelphia in the 1976. And today, to perform Legionella analysis, it takes around 14 days to obtain the results. So it's too late, if you have if you have high concentration of Legionella, you know it 14 days after. So during these 14 days, you will contaminate people, which is an issue. So if you are able to obtain a result only after 2 days, after collecting the sample, you will be able to adjust and to adapt and to react really faster in case of contamination. So it's why it's something very important. I hope that I have answered the question.
Yes, yes. Thank you so much. That was wonderful. I know we talked to Alex a little bit about some of the regulatory um, shifts that are impacting his work. Are you interfacing with regulatory authorities about the capabilities of rapid detection and how it may change sampling methodologies?
It's a very hot topic, because compared to chemical, analysis, microbiological analysis, it's a nightmare, To change the regulation, to change the rule, it will take years and years and years. So we need just to prove, that our solution is working very well. And when we are in microbiology, it's not simple to do it. So we are very happy because we have already several laboratories all around the world that have been certified, for our solution, which is ISO 17025, which is something that is important for people that are aware about microbiological evaluation. But it's not something that will change the game because they will not be allowed, to use our solution in replacement of the standard method. So the guideline will stick on the standard method that is coming from the 1997, if I remember correctly, for Legionella pneumophila. So, and there are no change since that time. So what we are doing is we try to educate the Ministry of Health. We try to educate the end user. We try to educate the laboratory to show them and to demonstrate them that we have a solution that is better or equivalent to the standard method in terms of reliability, but in the meantime, that will be able to reduce the time to obtain the result. And this will take a lot of time, and we expect that maybe in 10 years, we will be compliant, we will be equivalent to the standard method. So today, the people that are using our solution are using for their own purpose, it cannot replace the standard method.
Wonderful. Thank you so much for that insight. That's really helpful. So shifting to universal biosensors, I know you've been successful launching this product, but I know the journey to get to where you are was not easy. Could you touch on challenges that you've faced during the process of commercialization?
Yeah, absolutely. I think one of the main problems that we have found with sort of through the process of launching this product has been our inexperience in the water industry. We obviously, are very experienced in producing, rapid handheld products in a variety of industries, but have traditionally come from human health. So understanding how, the water industry in the US works, and then I guess starting to make the connections to help us through the development, but also move towards the commercialization has been really, really difficult. And things like working with CWA, going to some of the large industry conferences like the ACE conferences over the last couple of years have been really instrumental in us starting to make those contacts and then ultimately get into the position where we are now, where we have people in the field using it, trialing it, to hopefully finalise everything before we launch later in the year.
Okay. So we are going to start wrapping up with one of our last questions. So if you have anything else you want to ask, please use the Q&A function now. But Sam, you've come a long way on your journey as well. Could you speak to challenges you've faced in bringing your product to market? And are people skeptical about the abilities of devices capable of rapid detection when you tell them what you're capable of?
Yes, thank you for the question. It's very interesting. So yes, it's a long journey. And what surprised me the most, is that laboratory, that our main target are not happy, to change. And the question is why, why laboratory are not happy to change? And from my point of view is because they have the monopoly, you know, they have a business that is running well and they don't need to change, because they have customers that send sample and they have money for that. So now our strategy is to educate the end user, to educate, companies that have cooling tower, companies that have potential issue with Legionella, and explain them that we have a solution that will help them, that will help a lot for them, And by doing so, we expect that this end user will contact the laboratory and push them to start to use our solution. And by the way, I will have tomorrow a meeting with a big company in the US that perform a huge amount of tests. And when I start to explain them that, look, we can deliver the same result as the standard method, that deliver result in 2 days, in 2 weeks, just in 2 days, they say, okay, we will contact our contract laboratory and explain them that this solution is reliable, and we will make a meeting with them in order to use it. So we need more and more, end user that contact laboratory and say, look, here is a solution that we want to adopt, And by doing so, we expect that laboratory will adopt it faster. Sorry, Natalie, you are on mute.
Oh, no, you're all good.
Sorry. Okay. I thought that you asked me something, Yes. So the idea for us is here because people say, no, it's not possible. You cannot do it in 2 days. So, but when we ship them the equipment and we say, test it, they say, wow, we cannot see something in the membrane and we detect it. Or the membrane is crowded of microbes and it's not possible with our method, to do it. And with your method, you obtain the result. So they are convinced, but it takes a lot of effort,
Yeah, thank you so much for providing your input and how you're navigating this market. Are there any takeaways about rapid detection you'd like to leave the audience with today?
I think maybe.
Yeah,
well,
please
go ahead, Sam. Please.
No. So I think in the future, rapid method is essential because it will provide, faster result. It could be in chemical, it could be in microbiology, and you could adapt, your treatment. You could adapt and react earlier, and this will be essential for my point of view,
Yeah,
Alex,
I think
please.
probably adding on top of Sam there and about the struggles with the regulatory push, it really is going to take a massive undertaking from obviously innovators and companies, but also, as Sam said, end users to push the need for this across a number of different areas, to increase usage and then ultimately get regulators to understand how these tests work and that they can perform as well, if not better, than what's currently done, as a lot of the methods that are currently standard have been around for tens or decades or decades and decades of time. So I think that's probably the key to this whole area is getting people comfortable with doing things differently, understanding if there are differences, why there are differences, and that the results that they produce are just as good in terms of accuracy or reliability, and then have a number of other benefits which help everyone in the end.
Exactly.
Yeah. Awesome. Thank you both so much for joining us today. I really appreciate you being here, speaking to Universal Biosensors and Diamidex. I also want to extend our thanks to our sponsor again, Badger Meter, for sponsoring this Innovator Spotlight series. And finally, thank you to our network and supporters and everyone who participated in the Q&A. We really appreciate you being here and showing up to learn more about the innovations we see. And we hope to see you in July for our AI and machine learning session. And have a great day, everybody.
Have a great day. Thank you.
Thank you very much. Bye.
Thank you. Bye-bye. Bye.